Today's topic is about population genetics. Some people might consider this a slightly dry topic. It's more useful for people who are statistically minded and also work in the population genetics background. There is a lot of use in knowing population genetics, especially if you want to know what sort of diseases are prevalent in certain populations and what the genetic background for those people would be. So bear with me as I go through some of these topics, and we can always have further discussions if you have any questions later on. So population genetics is the quantitative study of the distribution of genetic variation in populations, including trends in the frequencies of genes and genotypes over time. So knowing about differences in allele frequencies across populations may be important to predict an increased likelihood of certain conditions in patients with specific ancestral origins. So what I mean by this is that in some ancestral origins, there might have occurred a particular variation, a genetic variation, or you could also call it a mutation or a pathogenic variant. And because populations stay together generally, the frequency of that particular variant is more in just people of that ancestral origin. So that's one of the reasons it's important to know, okay, if a particular person is of a particular ancestral origin, then there are certain genetic disorders that are more frequent in that particular population. It's also important to know that as humans, we all share an average of 99% of our DNA sequences across the genome. So this kind of variation is only a small proportion. So this is what happens with serial migration, a concept called gene flow. So gene flow is a gradual change in allele frequencies with migration or admixture of populations. So as migration of big populations have occur, if a particular gene variation was present in that population, it gets carried over to the other populations as well. So for instance, if there is a gene variant that was present in in the African population, as they migrate out to Europe, the African population carries that particular genetic variant with them to Europe, and hence the frequencies can change, and the gene keeps flowing from one place to the other. So that's the gene flow effect. This is slightly different to genetic drift, which occurs within a population when there is actually a random a genetic alteration that occurs. And particularly you find this in bottlenecks, when there is a very small population and a particular gene variant has occurred, or a pathogenic variant or mutation, however you would like to address it here. And that stays within that population. You wouldn't expect generally in that population for that frequency of a particular disorder, but because it's a very constricted small population, you could see that there is a higher frequency because of the genetic drift. The genes which are meant to be in that population are drifting into a different type of gene variant because of the constriction in the population. What is the founder effect? Founder effect is a high frequency of a genetic variant that originally occurred in a small ancestral group. So there might be that there is a particular gene variant that occurred in one or more founders who first carried this variant, and then it gets carried in subsequent generations. Founder populations with small effective population sizes are relevant for clinical genetics because individuals with these ancestries are generally at a greater risk of accumulating genetic diseases that are rare in other populations. As I was saying earlier, if there's a small population where there was a founder who started having a particular gene variant or a mutation, and then all of their offspring, and hence the offspring's offspring and anyone who partners within that small population can all accumulate the same type of genetic variant, and hence there might be a higher incidence of a particular genetic disease. For example, there is a high incidence of Huntington disease in Lake Maracaibo in Venezuela. It's a small population, hence there is less genetic diversity, so there are more people who are partnering with other people with the same type of genetic variant, and hence there is a higher incidence of Huntington disease in that population. Now let's talk about linkage disequilibrium. This is an important topic and many people get confused as to what linkage disequilibrium is. So I want to explain this to you using an example of, let's say, we take two bowls of colored sand. In one bowl we have pink colored sand, and another bowl we have green colored sand. And let's say we pour it all in another bowl and we mix it really, really well. So we mix it to the point of equilibrium where it's completely equal. Then you have equal amounts of green sand and pink sand in no matter what amount you pick up. If you take a teaspoon of that mixed sand, equilibrium sand, then you'll have equal amounts of pink and green sand. But if there is disequilibrium, which means it is not really mixed well, then if you pick up a teaspoon of that sand which has been mixed but hasn't been mixed really well, then you can see that there is a bit more green sand, the green sand sticking to each other, or the pink sand sticking to each other. So that's the concept of linkage disequilibrium. So basically, instead of the sand, if you just adopt the locus A and locus B. Say generally you would expect, if there is random mating everywhere and it's a mixed population, a completely random mixed population, then the A and B would be like it'll be all mixed really well. So some people would have there'll be A and there'll be another locus, and all of the loci get mixed up really well. But if there is linkage disequilibrium, then you would see that the uppercase A and the uppercase B stick to each other. So they stick together, and the lowercase a and lowercase b stick to each other. So instead of like similar to the pink sand sticking with itself and the green sand sticking with itself, the loci, uppercase loci, stick together, and the lowercase loci stick together. So this is actually the concept of linkage disequilibrium, which means it is not really mixed that well. So linkage disequilibrium is the non-independence of allele frequencies along segments of chromosomes that are inherited together. In other words, uppercase A and uppercase B are not independent. They're always occurring together unless there is recombination, which doesn't seem to occur. So if there's complete recombination of all of the sites, then there'll be complete equilibrium. But here what happens is that the uppercase A and uppercase B are always occurring together. Therefore, they are in linkage disequilibrium. So where uppercase A is present, there's also uppercase B. And where lowercase a is present, there's also lowercase b. So the haplotype uppercase A and uppercase B generally occur together, i.e. it is in linkage disequilibrium. When uppercase A and uppercase B don't occur together, they are in linkage equilibrium. I just want to introduce a few more terminologies. These terminologies are useful in population genetics. You may hear the term identity by descent, which is actually important in genetics. So what does identity by descent mean? When segments of maternal and paternal alleles are identical due to recent common ancestor. Say in a random population where the mother of a particular person and the father are completely random genetically, then you wouldn't have identical segments between the mother and the father. But if it is a small population with common ancestors, it is possible that the mother and the father have some segments of DNA that are actually common because they haven't mixed that much through outside populations, and hence it is likely that there is some identity in their genetic material because of common ancestors. And when the child is born, there is a chance that the child could inherit one segment from the mother and another segment from the father and have two identical segments. And the reason to know whether there is identity by descent, particularly with populations where people marry cousins or people marry within families and they have children, then there is a higher proportion of possible autosomal recessive diseases because the father might have been a carrier, asymptomatic carrier. The mother might have also been an asymptomatic carrier, but if the child inherits both those identical segments, then they've got one pathogenic variant from the father and another pathogenic variant from the mother, and hence the child can have a recessive disorder. And when there are lots of people within the population with this identity by descent, there is a higher proportion of certain recessive disorders. Genetically isolated communities have a higher proportion of identity by descent, i.e. because of genetically related reproductive partners. Another concept I want to introduce to you is called principal components analysis. It's a very, very statistical terminology. You'll only be coming across that if you're calculating polygenic risk scores or dealing with large population level data. So what is principal components analysis? It is a statistical method commonly used to separate population subgroups using genetic stratification. So there are different DNA markers for different ancestral populations. These are called ancestry informative markers, and statisticians are able to, just based on these DNA markers, separate out different ancestral population subgroups using this. So you can see in the figure here, each color represents a different ancestral subgroup, and these have been separated into different principal components. So you can see here PC1 and PC2, so two principal components using the DNA markers, ancestry informative markers, into different subgroups. Now, it's really important to know that geographic clusters, just because people live in a particular country, doesn't necessarily mean that they have the same ancestry. So geographic clusters are not necessarily similar to genetic clusters. Genetic clusters have common DNA markers, whereas geographic clusters are just a whole heap of people living in a particular place. So concept of minor allele frequency, this is also an important concept. So let's say that the different allele combinations in the entire population are PP, PQ, and QQ. So they're different alleles, right? So each of us has two alleles, one that we inherit from a mother and one that we inherit from a father. But the combination of that could be, varied. So some of us can have a combination of P from the father and P from the mother. Some can have a P and a Q, and others can have two of the Qs. And these are just allele combinations. These are not, the exact loci. So the allele frequency, if you're counting, how do you calculate allele frequency? It's basically counting all of the P alleles, including the PQ, and counting all the Q alleles. So if you want the P allele frequency, you count P in the homozygotes, and you also take into account the heterozygotes. The one that's PQ, you take 1. So you count the P's twice for the homozygotes, and you count the P once for the heterozygotes. Similarly for Q, if you want to calculate the Q allele frequency, you calculate the Q's twice for the homozygotes, and you calculate it once for the heterozygotes. One allele is likely to be more frequent compared to the other because we don't have complete thorough mixing anywhere. So we would always have one allele that is a lot more frequent and another allele that is not as frequent. And the less common allele is the minor allele. Okay. So estimating minor allele frequency in general population is useful to calculate the risk of having a recessive disease. So let's sample 10% of a particular population. So this is a sample population. There's a large population, 100%, but we're only taking 10%. And in this sample, in this 10% of the large population, it contains a 5% minor allele frequency for a biallelic autosomal recessive disease. So just name any autosomal recessive disease, and the Q allele is usually considered as a minor allele. So this allele is actually, codes for some kind of an autosomal recessive disease, let's say. And 5% of the alleles in this sample is the minor allele. So the frequency of the allele, Q, is 0.05, because it's 5% minor allele frequency. Now, since the trait has two alleles, whose frequencies sum to 1, so you know that we've got a P allele and a Q allele. So in a complete population, if you take all of the P alleles and if you take all of the Q alleles and add them up, it'll be, 100%. In other words, P plus Q equals 1. So you get all of the frequencies of all of the alleles if you just add up all of the P's and all of the Q's. So P plus Q is 1. Therefore, we can infer the other allele's frequency if we know one allele. So for instance, here we know that the frequency of the Q allele is 0.05. So what is the frequency of the P allele then? So P allele then is basically 1 minus the Q allele, right? So the other allele's frequency is 0.95, 1 minus 0.05, which is 0.95. So now we know the P allele frequency based on the Q allele frequency. We calculated that the Q allele frequency is 0.05, and now we've also, based on that, calculated the P allele frequency, which is 0.95, because the rest has to be the P allele. So now we only sampled 10% of the population. Let's apply this to the entire 90%. So whilst this small population has the Q allele of 0.05, because they've got a high incidence of an autosomal recessive disease, the rest of the population does not necessarily have a high incidence of this autosomal recessive disease. So the Q allele, the allele that carries the disease, is negligible when you consider the entire population. So if you apply to the entire population, only this population's got a high recessive disease, but there might be carriers, which means that there could be PQs, right? So in the remaining 90% of the total population, the frequency of this pathogenic variant is so small, it's negligible, that it is generally not observed. So we don't really see the Q allele as much in the general population. It's special for this particular small population with a high incidence of a recessive disease. Therefore, it is presumed to be absent, such that in the entire population, we're not talking about this 10% sample that we took, in the entire remaining 90%, we assume that Q is 0. Therefore, if Q is 0, P has to be 1, approximately, of course. So in this case, you can counsel your patients that if they partner in that 90% of the general population, then the chance of them having a child with one abnormal allele from them and another abnormal allele from the other parent, so having two Q alleles, is negligible, because we believe that the Q allele is really, the chance of the Q allele being there in the remaining 90% of the population is negligible. Therefore, the chance of them having a child with two Q alleles is going to be negligible, even if they are a carrier themselves. Does that make sense? Now, there is a difference between allele frequency and genotype frequency. For autosomal loci, the possible genotype combinations in the population, assuming the alleles are P and Q, so the alleles are P and Q, but you need to have two of these in order to form a genotype. So the genotype could be PP or PQ or QQ. So how do you calculate the allele frequency of P? So you calculate, as I was saying earlier, 2 of the individuals who are homozygous for P and 1 for each individual who is heterozygous for P, and divided by 2 times the total number of individuals in that population. So that's how you get the allele frequency of P, and the same thing for Q. For allele frequency of Q, you calculate 2 times the number of individuals who are homozygous for Q and add that to 1 time times the number of individuals heterozygous for Q, and then divided by 2 times the total number of individuals in that population, and then you get the allele frequency of Q. Another concept that is actually important to understand, especially if you're dealing with population genetics or large databases, is Hardy-Weinberg equilibrium. So if you're calculating things like polygenic risk scores or dealing with disease frequencies in large populations, then Hardy-Weinberg equilibrium is really important to understand. So if we know the genotype combinations, so PP, PQ, and QQ in a population, we can calculate the allele frequencies, right? So we can calculate the way we just showed. So we can use 2 P's and then add it to 1 P, which is for the heterozygotes, and then divided by 2 times the entire population. But can we do the reverse? Can you calculate the genotypes? So how many people are PPs and how many people are PQs and how many people are QQ if we know the allele frequencies? So if you just know that this is the number of Qs, this is the number of P alleles. So this is not easy without knowing how many homozygotes and heterozygotes are there for the alleles. If a population meets certain assumptions, an equation called Hardy-Weinberg equilibrium can be applied to calculate the genotype combination. So we'll talk a little bit more about Hardy-Weinberg equilibrium. So there are some assumptions that need to be present before we can apply Hardy-Weinberg equilibrium to any population. So the principle of Hardy-Weinberg equilibrium rests on the assumption that genotype frequency proportions remain constant over time because of the following. First, that there is random mating. Reproductive pairings are random with respect to the locus in question. and there is no genetic drift. We talked about genetic drift earlier. The population under study should be sufficiently large such that alleles are not likely to dramatically rise or drop in frequency by random chance. So if there is a particular variant or a mutation that occurs, it's not going to be inherited by all of the offspring because it's a tiny population with just a small number of founders, and then everyone will end up having that particular variant or mutation. So there shouldn't be a genetic drift, and this phenomenon, genetic drift, can be avoided if the population under study is a large population. There should be no mutation. Rate of mutation is low, such that allele frequencies are not impacted. Even if there is a mutation, it's very small amounts, so it's not affecting the entire population. And there should be no selection. Individuals are equally capable of passing on their genes regardless of genotype, preserving equality between each allele frequency and its chance of being inherited. So an example of selection could be that, say somebody's got a genetic disorder that is so severe that they cannot actually reproduce, then that particular allele just dies out. Yeah. And also no gene flow, that there's been no significant migration of individuals between populations with significantly different allele frequencies. So if these assumptions are all met, then we can apply the Hardy-Weinberg equilibrium to a large population. So let's have a look here. This is pretty simple for you to follow. So if you can look at the parental genotypes, so uppercase A, and then if there are two parents with both uppercase A genotypes, then you can see what happens in the F1 generation. Now, there are different combinations that you would see. If both have uppercase A's as their genotypes, then the children will only have uppercase A. But if they have a combination of uppercase and lowercase, then you can see the different combinations there. And you can see how the populations eventually start having different combinations of genotypes based on their parental genotypes. So now the offspring genotype frequencies. So let's start talking about, from the just genotypes to genotype frequencies given parental genotypes, assuming Hardy-Weinberg conditions. That is, there is no mutation. So you can see here, so there's two uppercase A's and then two uppercase A's in both parents. So you can see that that all the children, the genotype combinations will only be uppercase A. Now let's say there's one uppercase A and one lowercase a, and the other parent also has one uppercase A and one lowercase a. You can see the combinations here are varied. So there is 0.25 chance of two uppercase A's and 0.25 chance of one uppercase A and one lowercase a, another 0.25 chance of one uppercase A and one lowercase a, and another 0.25 chance of two lowercase a's, right? So if you look at all of this, then we can actually start applying the Hardy-Weinberg equilibrium equation based on this. So have a look at this in more detail. It may be a bit overwhelming when I'm just talking about it, but take a look at it at your own time, and you'll recognize that there is a pattern here, and that there is, even though there is homogeneity, there is a pattern based on different genotype combinations. So based on that, we can actually derive an equation for Hardy-Weinberg equilibrium. So the Hardy-Weinberg principle states that the frequency of the three genotypes, so uppercase A's, or uppercase A and a lowercase a, or lowercase a's, is given by the terms of the binomial expansion. So instead of uppercase A, we apply P, and instead of lowercase a, we apply Q. So if P plus Q, we've learned previously that P plus Q is 1, right? So if P plus Q is 1, P plus Q the whole squared should also be 1. So we're talking about population frequency here. So if you apply the algebraic expansion of P plus Q the whole squared, then we know P plus Q the whole squared is P squared plus 2PQ plus Q squared, right? So these are the different combinations, genotype combinations in the entire population. So you'd have a proportion of people who are P squared, a proportion of people who are PQ, but there's double, there'll be double the amount of carriers, right, 2PQ, and there'll be a proportion of people who are Q squared. And if you add all of these combinations together, then you get the entire population. So P squareds and double the amount of PQs and Q squareds together makes the entire population. In other words, p plus q the whole squared, also known as p squared plus 2pq plus q squared, is all equal to the entire population, or a proportion of 1. So is whole. So Hardy-Weinberg equilibrium applies to autosomal loci and X-linked loci in females, but not in males. So applying Hardy-Weinberg equilibrium to autosomal recessive traits. So the major practical application of Hardy-Weinberg principle in medical genetics is in genetic counseling for autosomal recessive conditions, to counsel about risk of having an affected offspring. So you may see patients who say, hey, I've got a brother who's got phenylketonuria. What are the chances that I might have a child with phenylketonuria? I don't actually have the condition, but do I have like a hidden gene for it or something like that? So let's take an example of phenylketonuria. Assume that the frequency of pathogenic alleles in the population is Q. Therefore, the frequency of non-pathogenic allele P is 1 minus Q, assuming Hardy-Weinberg equilibrium, right? So if we were to attempt to determine the frequency of disease causing PKU alleles directly from genotype frequencies, we would need to know the frequency of heterozygotes in the population. In other words, we need to know the homozygotes P square and Q square, as well as 2PQ, the heterozygotes. But heterozygotes are asymptomatic silent carriers, and their frequency in the population, which is 2PQ, cannot be reliably determined because they're just asymptomatic. So they could easily be P squared. We don't know if somebody who doesn't develop phenylketonuria, are they P squared or are they PQ? We don't really know. So we can't easily determine directly just from the phenotype, just by observing them. So now let's apply that. So we've got Q and we've got P, and let's apply that to genetic counseling in PKU using Hardy-Weinberg equilibrium in a particular population. Let's say frequency of PKU in a population is 1 per 4,500. So 1 per 4,500 babies born in that particular population have PKU. Then we know that the frequency of affected individuals, Q squared, is 1 per 4,500. So Q squared is 1 per 4,500. From this we calculate that Q, so if Q squared, so square root of that is 0.015. So Q is 0.015. So if you know Q, now we can calculate P. So P is 1 minus Q, so 1 minus 0.015, which is 0.985. So now we've got Q and we've got P. So we can easily calculate 2PQ, which is the carrier frequency, right? So 2PQ in this case, 2 times P, which is 0.985, times 0.015 is 0.029. So this is the frequency of asymptomatic carriers in that population. So it's approximately 3%. So if a known carrier, so say somebody, we've done genetic testing, we found that they've got 1 Q allele and 1 P. So we know now that this person is a carrier, but they say, hey, who should I partner with if I want to not have a child with PKU? How do I know what their genetic combination should be? So if a known carrier partners with an unaffected person from the same population, then we just say in this population, any random partner has approximately 3% chance of being a carrier. So you give them the estimate based on what we've calculated. Say that, hey, there is a 3% chance that your partner could also be an asymptomatic carrier. And based on this, they can make their decision, oh, well, it's only a 3% chance and that's okay. I'm happy to partner and try and take my chance of having a child with this person. Or they might say, no, I do not want to have a 3% chance of having a partner, so I would choose a partner from another population. So this estimate can be used to provide genetic counseling. Or they might wish that their partner should be genetically tested to also find out. And if there is a high frequency, so if it's more than 10% in some places, or depending on the chance of their partner being a silent carrier or an asymptomatic carrier, we could also perform genetic testing. If there is a high chance, and the chance of us using that money to do genetic testing for them is justified. But say if partners only got 2% or 1% chance or 3% chance, and if they go to a genetic center and they say, hey, I want my partner to have genetic testing, it may not, we could actually advise them that it's not the wisest use of spending money, because the chance of us finding anything in that particular person is not very high. Now lastly, I just want to talk a little bit about genomic variation and biases in population data sets. So there are population reference data and genomic databases, so they collect a lot of information, genomic information from different populations. Anyone who's had any kind of genetic testing or genotyping, a lot of populations have participated in that, but it's heavily biased towards a European genomic background. This ascertainment bias towards European ancestries contributes to higher rates of variance of unknown or uncertain significance in non-Europeans. Say, for instance, someone of an African ancestry comes to you and they have a particular genetic variant. They have a heart disease, a genetic heart disease, and we do test their genes, and they've got a particular variant. And you look at the European databases, and people with that genetic variant actually, do have a genetic heart disease. Let's take an example of hypertrophic cardiomyopathy. And then we would say, yeah, you've got hypertrophic cardiomyopathy gene. You've got the genetic problem for hypertrophic cardiomyopathy. So this is actually something that happened in real life. There was a pathogenic variant associated with hypertrophic cardiomyopathy in European population. But then there were people of African American origin, and they actually ended up having the same genetic test, because there was some cardiac problems in their families as well. And they were told that this can actually cause heart problem in their families. So many of them ended up having preventative cardiac procedures if they had this gene variant. But later on it was found that it is not the case in African populations. It's only the case in European populations. So the real cause was missed, and lots of people ended up having unnecessary cardiac procedures because this particular pathogenic variant or mutation only applies to the European population and not to the African American population. Several efforts are currently underway to improve diversity and inclusion in genomic databases, so watch this space. There is a lot of effort that is happening, particularly the African population as well. So it is possible that in the future we would have a more homogeneous mix of different databases instead of the way it is, as you can see here, heavily biased towards the European population. So with that, I would end this particular lecture, and I'm very happy to take any questions if you've got any.